Thank you so much, Andrew. And again, if anybody has a question, go find Andrew at his poster just after the other two flashcards. Now we have another ECR, Dr. De Angelis, also from the Roslin Institute.
Hello, UK-ICN. Well, first of all, thank you for being here to the last day and surviving till now. Today, I'm going to try and decode with you the hidden message of the coronavirus nucleocapsid gene. So in coronaviruses, the N gene of many coronaviruses actually has been found to encode an accessory operating frame in its second reading frame, such as SARS-CoV-2 with ORF9b, Porcelain Delta coronavirus with S7, MERS-CoV with ORF8b, and so on and so forth. And at the beginning of my PhD, I kind of wanted to know, well, how common is this feature across coronaviruses? So I took the N gene of 136 different coronaviruses and I started mapping operating frames across their alternative reading frames into maps like this. So you see, this is for a beluga whale coronavirus. I know, poor beluga whales. And they have on frame one long operating frame that starts with a start codon here in red and a stop codon here in black. But if you look at the second reading frame, it's full of very small ORFs, lots of start codons and lots of stop codons. But if you look at frame 3, they're actually full of stop codons. You don't really see anything. And this is a common pattern I kept seeing across all coronaviruses, or most coronaviruses in my data set. And I kind of wanted to know why that was the case. Why we're seeing this uneven distribution of stop codon, start codon, and so on and so forth. And so what I did, I basically did a codon composition frequency analysis across all 3 reading frames, across my whole data set. And in frame 1 here, you see from the x-axis, you have the most common codon that you find in N on average across all coronaviruses. And here at the end, you have the least common codon, which are stop codons very luckily, because I mean, that's what you would be expecting. But if you move on frame 2, you actually see that there's a massive overrepresentation of codons such as ATG, CTG, and also GTG, which are potential start codons in alternative reading frames. And if you look at frame 3, you actually see that there's actually quite an overrepresentation of stop codons. So that's why we're seeing this pattern. But I was interested in seeing how this codon pattern could affect whatever we were seeing, whatever ORF we were seeing in the alternative reading frames of all these coronaviruses. So I took the N gene of 5 different Mirbeco-viruses. I stuck a transcriptional regulatory sequence upstream of each N, added a flag tag at the end of each ORF8b gene, took these sequences into mammalian expression vectors and transfected into cells. And when I looked for these proteins through Western blots, I actually realized that I could detect multiple protein products for the majority of all these ORF8bs, from MERS-CoV here to Hegio coronavirus ORF8b. So this kind of suggested the expression of most ORF8bs leads to the production of multiple polypeptide species that could possibly be expressed by the multiple start codons that we see within these ORFs. Now, I wanted to, being able to express them, I wanted to see the differences between these proteins. So I wanted to see what they were doing inside the cell. So I went to confocal microscopy and, right. Sorry, people. You didn't pay for the subscription. So if you really want to know more about it, you kind of have to come to my poster, poster 8. Right. But thank you very much.
Fantastic way to stop. Thank you very much. Great stop code on this.